5 minutes. Yep.
5 more.
Yep. All right. Why is this? Oh, there we go. All right. The subunits in the holoenzyme for RNA polymerase in prokaryotes. So, do you guys know? This one is the hardest one, and it's a sub note in her notes, and it's in the book. So, just take a guess. I'm just gonna I'm just gonna throw out that this one is the hardest of them all. It is Greek. So it's alpha, alpha prime, beta, and sigma prime. Alpha, beta, sigma, sigma prime. Alpha, beta, beta prime. So it is two alphas, a beta prime, and a sigma. And that's your sigma factor is in there. So it was like a tiny, tiny thing. So of course I knew that one. Which will cause bacteria to become resistant to erythromycin. So, what happens to erythromycin? So, she mentioned this in class, and I think that she has it in like her sub notes. So, so that's the structure of erythromycin. Okay. So, if you guys noticed a second ago, it's like a big ring. So, what happens with erythromycin? It has all those OHs all around that ring. The there's like a hydroxylase or something, and it like pops apart that ring. And then once it pops apart that ring, it doesn't work anymore. But do you guys know where on what subunit erythromycin goes to? I believe so. Yeah. Where does erythromycin go to? The 50S? Yeah. Oh no, that's why I'm doing that. All right. Which base pair binding is stronger? The T to A or the C to G? Easy one. Yep. Why is that? Three. I'm at the top. All right. Which nucleic acid base is methylated? Okay. So thymine looks almost exactly like uracil, but it has a methyl at the bottom of it. I think that was one of your questions. No? Oh, that was one of yours. Yeah, I wouldn't have thought of that, but yeah. Oh, I tried to purposely misanswer that one. All right, which amino acid is the limiting reagent in DNA synthesis? She mentioned this in class. I think I misspelled. All right, so it's thymidine. So, um,
It's, I think it has some names. Hamidine something.
what?
I think the name was Hamidine.
So it's the triphosphorylated one. Yeah. And the reason is that those are used in different processes in the cells. So different ones. And so that one just gets used all the time. And so it's always hard for the body to. So what can happen is that you can make a specific target for thymidine and inhibit all the protein synthesis, DNA synthesis by just targeting that. So that's why it's important with medicine, maybe, because you can. No, no, this is just the A to T binding. That's all it is. So this was in that table. So what process does streptomycin interfere with? And that is the structure of streptomycin. So if you guys notice, there's like a little sugary looking thing in there, and then there's things that look kind of like bases in there. And so that's why it kind of messes up with stuff. What? Oh, this is on the stuff from today. Yeah,
Mhm.
yeah. It's on the like second.
Carbamazine, tetramazine.
Yeah.
Mhm.
Yeah, yeah. No,
But I don't think she had the structure.
no, the structures, the whole entire reason I'm showing you guys the structures is just so that you guys can recognize they look like bases. Because if you look at them, they look kind of like bases. That's all the reason I'm showing you them.
Yeah, but on the exam, it will be something like, yeah, that is, and also what is binding to like what I did, amino or tRNA and those kind of things.
Well, she surprised us today, so
Yeah.
she surprised herself today.
Yeah.
So, but yeah,
Today's quiz should have been like everybody 100. Period.
all right. Why is telomerase not used as an anti-aging therapy? She has this in her notes. So, so is it only present in stem cells? Is it not present in humans? Does it cause cancer? No, it is a therapy. You're wrong. So, she kind of mentioned this. It can cause cancer. So that's the development hiccup. But once we can figure out how we stop cancer, then we can be immortal. So, hey, people. All right, where is the poly A on the mRNA? Is it at the 5 end or the 3 end? It's at the 3. What direction is DNA synthesized? So some straightforward ones. Make you guys angry with mine. So it is high to low. It's 5 prime to 3 prime. I got kicked out of my own thing. All right, what is the job of the Golgi apparatus in the cell? So does it make systolic proteins? Does it produce proteins for export? Does it produce cellular lipids? Or does it just export proteins? Okay, so it just exports proteins. Which one produces the cellular lipids? Smoothie R. Which ones produce the proteins for export? Yeah, so the rough ER allows the ribosomes to just straight up produce the protein into a vesicle, and then that goes over to the Golgi, and then the Golgi attaches like tags essentially sometimes as to where it should go. So we sometimes call those postage stamps, but good. Nobody systolic proteins. What is that? That's your free ribosomes. So she has that in her objectives, so I put that in there. How many base pairs per turn does DNA have? So we're going to assume the B conformation. So I have the B conformation up there. And she has this in her objectives, but I don't remember her ever mentioning it in class. Yeah, so, and I couldn't. So, yeah. So, all right, this is my problem with it. So I was taught in the crystalline form, it's 9.8. And then in the hydrolyzed form, it's 10.5. But her notes say 10. Yeah. So I have 10 marked as right. So we're just gonna go with her notes, but that's why if you end up in another biochem class someday no, you never will. But Which form of the DNA is the crystalline form? So which one is not hydrolyzed? Which one doesn't have water in it? So A is the one that doesn't have any water along its main groove, and then B is expanded out and it has water along its main groove. Z is the one that is completely the other way. It's going the other way. And it's might have water, it might not, but she doesn't have it in her notes. Okay. Well, then that leaves only A then. But the thing is, it's too expanded out. I thought that it was too expanded out to really be crystalline. Hmm. I don't know. Um, Rosalind. Yeah. All right. GTF works with all RNA polymerases. True? False, only polymerase 1. False, only polymerase 1 and 2. False, they are polymerase specific. GTF. She has this in her objectives that we need to know what GTF is. And she has it confusing, and that's why I specifically made this way. So, what does GTF stand for? It's like general transfer. What? Yeah, transcription factor. And that, yes. And then it binds with the polymerase, the RNA polymerase. But she has in like a side note that it's specific to each polymer. Yeah, that I'm kind of getting a little bit peeved about the like little side notes. And then she has it in her objectives that we have to know her side notes. Um, I'm gonna guess it's just gonna go. Yeah, yeah. That's your, what is that, protein initiation complex? Yeah, or polymerase. No. Yeah. A type of terminator that involves inverse repeat forms that destabilize the DNA RNA hybrid. So, so which terminator destabilizes it? So this is one I pulled from the book. So the ones that involve the inverse repeats, what basically happens is RNA doesn't stick quite well to DNA as much, and it's partially because of uracil. Um, and so eventually it has these little codons that are inverse of each other. So like one will pick up and then they'll go and then just rip off. So it's like seeing like a bunch of magnets coming off and then sticking together. So it's interesting. What? No. Polycistronic is something completely different. We'll get to that. Oh no, we're having tricky questions tonight. Nope, this is why people should make their own questions today. Because I will. Which nucleoside is associated with the methyl cap on the mRNA? So, which one? Which one is generally the one that is methylated? And there's a reason for that. Is because it's just there's an enzyme for it that's pretty abundant. But she has it in her notes, and somebody else. I think you wrote this. You wrote it. Yep. So why would you have a methyl cap on an mRNA? It helps keep it from degrading, and it makes it stable, and you can splice it easier. So, um, I think so. I think it does. I am not quite sure because she doesn't have a lot of stuff on this as to like what we need to know. So, but the stuff that she does have, it's like side notes. So, why is the difference in ribosome sizes between eukaryotes and prokaryotes significant? So, and this was from, I think, the pharmacy law. After the pharmacy law, I did some biochem review. So this is one of my questions from that. So if you get it right the first time, you get it right this time. Okay. So the differences in the ribosome sizes, the reason they're significant is because it makes it a good drug target. That's why it makes it good for the antibiotics. Because one will specifically bind to one ribosome and not the other. Which way does prokaryotic DNA synthesis occur? Okay, I did grade this right. All right, yeah, I think it goes the same way as the eukaryotic. This is a trick question. But in her notes, she has like it's red 3 to 5. She has something where it says it's red 3 to 5, but it's really not. It's still synthesized 5 to 3. Yeah, so she has it confusing in her notes, and that's why today somebody asked about that, and so I was like, that's a good Kahoot question. Make sure everybody. All right. During which phase of the cell cycle is DNA replicated? Is it the mitotic phase? Is it the growth 1 phase? Is it the, I think it's, I don't know what the S phase, but then there's growth 2. But the other thing is if I say the S phase, I think you guys will. Okay, so pretty much everybody, so that's the synthesis phase, which, yeah, that's why I was like naming them off, and I was like, oh no, never mind. But yeah, she has that in her objectives in the Word doc is know this. Hey people, okay. What is required by RNA polymerase 2? So this one I pulled out of the notes. It's a little bit funky. So this is another one. This is the GTF again. PIC is what GTF and POL2 together are.
But it required because you had your GTF plus your polymerase. It's going to give you your PIC. So the PIC requires the GTF and the polymerase too. So I don't know if you, because if you say 2 plus 1 equals 3, to get 3 you need 2 plus 1. I don't know. So you get
It's kind of a technicality. So
yeah. Yeah. So PIC is what required.
it makes PIC.
It makes PIC,
GTF.
but it needs GTF to make PIC. So
Phase, but it's fine.
I know it's a bit of splitting hairs.
It's good, you
I
can
know.
do outside the box.
But hey, that's what today was splitting hairs in her quiz. I was like, where does the production of ribosomal RNA begin? So, all right, cool. So, nucleus, nucleolus. So yeah, the cytoplasm would be right if it was like a prokaryote, but I believe I was kind of implying by the answers that it was a eukaryote. I probably should.
Well, like I said, I did study you prepare something. Saw you.
Yeah. Which polymerase is in the mitochondria? So is it beta, delta, epsilon, or gamma? So we have 5 eukaryotic different ones. So I don't have alpha in there. So you obviously know that one's not in the. So it's the gamma. So everybody pretty much got that. Yep. So there's only one that's in the mitochondria. Where are the rest of them? Nucleus. Okay. All right. True, false. In prokaryotes, the transcription and translation are coupled, so they happen at the same time. Yep. Everybody got that. I purposely answered false. All right. Uracil binds to. All right. Everybody got that. So what would adenine bind to instead of uracil if uracil wasn't there? If this was DNA. All right. All right. Post-transcriptional modification can only happen in eukaryotes. So yeah. So it happens both in eukaryotes and prokaryotes. I thought that that one would be tricky because it's like one bullet point in her thing, and she kind of emphasizes it in eukaryotes, but it can happen in both. No, there's not that, but you can have acetylation, you can have methylation, you can have palmitoylation. What's another one? She had a whole bunch of them, but yeah, yeah, but that can happen in both. What is the type of bond between the histone and the DNA? So, yeah. Yeah, I asked this one last Kahoot. Yeah, so it's an ionic bond. It's not a covalent bond. Um, yeah, I thought that that was tricky, but she had it on her objectives. I was like, all right, then I don't have to take out this question. Awesome. All right, tRNA has the code. No, it has the codon. Sorry, it's not supposed to have a. So trick question is an anticodon. It's not the codon. The codon is on the DNA. Anticodon is on the tRNA. All right. This one has multiple right answers. So, what are polysomes? So, are they GTF enzymes? Are they activated ribosomes group? Are they ribosomes bound to mRNA? Are they polymerase digesting enzymes? So you got a 50-50 shot because there's multiple answers to this one. All right. So in her notes, she has them as an activated ribosome group. So, because most people didn't get that one. But in our book, it says that it's activated ribosomes bound to mRNA. So we're going to go with her notes and say it's a ribosome group, which makes sense because they kind of have like van der Waal interactions and they kind of like just drift towards each other. So depending on like where they are in the cell. But yeah, you can end up with like a huge conglomeration of ribosomes just kind of chilling out with each other. Eukaryotes mRNA are polycistronic. True, false, they are monocistronic and polycistronic. False, they are monocistronic. False, neither. You made that up. So yeah, which ones are polycistronic? Prokaryotes. So what happens in eukaryotes is the SNRPs that she likes talking about take out the introns, if I remember correctly. I keep mixing up introns and exons, but yeah, introns takes them out, puts all the exons together, and it just codes for one amino acid. That's it. Whereas with bacteria, their genes don't usually have introns in them, first off. And then, second off, because they have their mRNA transcription and translation at the same time, they don't have time to splice and do all that stuff. So they just go from one to the other. So if that helps, does that help people remember this a little bit better? Okay. Hello, people. No. How long is DNA per every turn in angstroms? She has this in her objectives. I don't remember seeing this anywhere in the notes, but I knew this from my biochem class. So I don't blame you if you don't get this right. So yeah, it's 35 angstroms. Well, it's 34 technically, but of those that's most correct, it's 35. Because I don't want to put like 25, 30, 34. Kind of, you're smart kids. You'll use the exam to figure out which one's right. Which will cause bacteria to become resistant to tetracycline. That's the picture tetracycline. So she talked about this in the class about how it mutates something. So that should help you. Yeah. It mutates the 30S unit on the ribosome, and it's one of the few where the prokaryotes will directly just mutate something in order to not bind. So if you guys saw like all those rings, why it's called tetracycline, it's 4 rings. Ah. So we're clever. All right, what is the job of the protein cyclin in DNA synthesis? So cycles DNA into the chromosomes, it starts the DNA replication, signals the beginning of DNA synthesis, and cycles ribosomes out of the cell. So this is another one where she has one bullet point, but then she has it in her objectives. So yeah, it is in the part where it has the circle with the, I think the G, the M, G2, S. So that, and then she has the cyclin-dependent kinases. So the cyclin-dependent kinases, you guys also have to need to know, which she has in her objectives. So they are phosphorylated by the cyclin. And the cyclin, once it gets phosphorylated, will start the beginning of each cell cycle. So there's different cyclins, and each cyclin starts. So like one starts G1, one starts M, one starts S, one starts G2. But she has in her notes that it starts DNA synthesis, which is kind of true. There's one cyclin that will. So that's, anybody mad at me about that one? Okay. Hopefully, like this one I thought was like a super tricky question, but most of you guys got it. So did somebody get kicked out? I'm seeing 6 people. Oh. Which form of DNA is the most common? So easy question. Yeah. Everybody got it. Yeah. Which part of the mRNA will dictate the stability of the mRNA? So is it the leader sequence, the coding sequence, or the trailing sequence? And she like highlighted it up on the board, so I have it highlighted in my notes. And then she's like, know each one of these sequences and what they do. It's like one slide specifically, and I was kind of a bit peeved. So yeah, evidently it's the trailing sequence. I didn't know that before today, but we learn things new every day. So it's evidently that poly A on the end. That evidently dictates way more stability, which makes sense in some ways than the methyl at the beginning. So the methyl will like kind of prevent some degradation, but that poly A at the end will really make it nice and stable. So, but she has that in her objectives too. So we're all learning things. Where does the polymerase alpha work and what does it do? This is why polymerase alpha wasn't in the one with the mitochondria. So, is it in the nuclear? Does it do DNA replication? Does it do DNA repair? Does it do DNA proofreading? So, yep. So, it is the one that does replication. So, which ones do repair and which ones do proofreading? Do you guys know?
Beta and delta do, or not delta, beta and delta have one for free. And I don't know, whatever mitochondria does.
Mitochondria, one's gamma. It does everything. So, what's interesting about the one polymerase gamma is it is almost identical to the one in prokaryotes because prokaryotes have one. So this is the kind of theory that we swallowed a bacteria a long time ago and turned that into our cellular powerhouse bitch and made us, it enabled us to be strong people because we literally have little bacterial slaves in our cells. How do aminoglycosides work as an antibiotic? So, here's another one that looks like a lot of rings and such. So, does it cause mRNA misreading? Does it cause early translation termination? Does it inhibit the DNA synthesis or does it inhibit the RNA synthesis? So, all right, so this one, it binds into, if I remember correctly, I think the amino acyl pocket on the ribosome. And so it tries to bind it into the peptide chain. And so it just clogs up the works on the ribosome. So it causes a whole bunch of problems. All right, how do eukaryotes replicate all of their DNA in a reasonable amount of time? So they move at 10 kilobase pairs a minute. They replicate continuously throughout the cell cycle. It has multiple origins of replication, and eukaryotes have less DNA than prokaryotes. if I was a professor,
That's my question.
that wasn't my question. see, this is why, because I will do trick questions, but so the DNA polymerase moves at 10 kilobase pairs per minute. Yeah, it doesn't move that quickly, but the problem is even if it did, it would still take it like a week at that rate, whereas at 2, which is the actual speed, it takes it like 35 days. She has that as like a side thing. The DNA replicates continuously throughout the cell cycle. No, because then it would be exposed to problems. It wouldn't be able to separate. You would have your mitotic phase would be all messed up. Eukaryotes have less DNA than prokaryotes. Which do you think is a more complex creature, humans or bacteria? Just that's a way you can think about it. So there's obviously more DNA in a eukaryote than a prokaryote because prokaryote has the little circle. That's it. But yeah, so what happens is that the DNA is just multiplied at multiple sections. And so she has a picture of like the little bubbles in the DNA coming out. And so eventually what will happen is that the DNA polymerase will hit the end of a RNA primer. It'll go boonk, fall off, and then a exon nuclease will come by, take out that RNA primer. Another RNA or DNA polymerase will come in, put in the right DNA, and then a ligase will come by and just basically tapes it together, the DNA. So I imagine all DNA synthesis as arts and crafts with children. How many prokaryotic polymerases are there in the cell? Maybe, maybe not.
So like total polymerase
No, there's just one.
or it's RNA.
So it's prokaryotic, it's bacteria. Bacteria only have one.
Yeah, they have
I thought,
one RNA.
I thought that. Oh, you know what? I didn't specify. You know what?
I think one. 'Cause
But-
I thought RNA first stuff up for
Yeah, yeah. So, yeah, eukaryotic. I think it's 3. Yeah. You know what? Usually catch this stuff on myself. I'm not catching stuff today. In RNA or RNA, what is modified after synthesis? So this is a question from last time, that there's only one thing that gets modified on the rRNA. And she mentioned this in class. It's in the first nucleic acid thing. So this is an old question, but it's useful. It's in her objectives. So it's just the base. Just the base gets modified. Phosphate can't really get modified. There's not really much to modify. The sugar could technically, but it doesn't for some reason. So what does chloramphenicol do as an antibiotic? So does it bind to the aminoacyl site? Does it bind to the peptidyl site? Does it bind to mRNA, or does it bind to the tRNA? So it binds to the aminoacyl site. So do you guys know where the aminoacyl site is? It's the first one. So peptidyl one is the one where the peptide bond happens. Are there protein synthesis inhibitors in eukaryotes that are used for therapy? Yep. So when would you see a protein synthesis inhibitor? Do you guys have an idea? Nope. Good guess, though. Cancer. So you want to stop cancer from growing. So we give it a protein synthesis inhibitor or a DNA inhibitor. And usually tumor cells grow at a much faster rate than everything else. So your fastest growing cells end up getting affected, which is your hair, your nails, your skin, lining your gut. So those ones end up, that's why cancer patients look like, well, they look like cancer patients, is because they lose their hair because that grows fastest. They start to have digestive problems, all that sort of stuff. So it's useful to know this because we'll see it again later in medicinal chemistry. All right, so this one I believe is hard, but what is the template-directed synthesis of RNA, which is the negative 35 base pairs downstream? So yeah, it's the cat box, which is that one. The Tata is the one underneath it, that yellow. That one is 10 base pairs. But those are the two that you should know because she highlights it. But I don't know. I don't know if that's going to be that specific, but it is highlighted. So why is the maturation of mRNA important in eukaryotic cells? So is it so degradation can occur? Is it to prevent degradation? Is it to start translation, or is it to stop translation? So yeah, it's to prevent the degradation of the mRNA so that you don't have any errors happening in the proteins. No, what is this? So can you identify which one this is after your brain's been liquefied a little bit? So first off, is this a purine or pyrimidine? I believe so. Okay, so this one is cytosine. So thymine, uracil, and cytosine have one ring. Adenine and guanine have two rings. Thymine has the little methyl at the bottom. Uracil has two OHs, or well, H's. No, O's off the side. Yeah. What? Yes. Yes, because it's based off of yeah. I just remember that the bigger the thing, the smaller the label, and the smaller one has the bigger label. So it's inverse. Yes. I don't know why I was sitting there. I was like, I'm dead. I'm going to go sleep after this. Which antibiotic is a reversible binder? It's the only one that she had in her notes that was a reversible binder. So the reason that we use tetracycline, erythromycin, and aminoglycosides a lot is because they stay on the binding site and they don't come off. Whereas chloramphenicol we don't use as often because it's a reversible. So you have to have a high amount in order to have the antibiotic effect. So, but she has in her notes that this is a reversible binder and that's all. So, but it's the one distinguishing thing about it. Last question. Yay! Which protein synthesis inhibitor can affect both eukaryotic and prokaryotic cells? So there's two of these in here. What?
I'll go with one of them. Sure. There you go.
All right. So erythromycin just does prokaryotic. Cycloheximidine, sorry. That one is a poison. It just does eukaryotic. And then puromycin evidently does both, and tetracycline does both. So tetracycline is actually really, really reactive. It degrades quite quickly. So yay. Whew. So you guys good? Questions? Hello, people? No. I don't think people are awake until that sound.